Okay, so in the summary, so according to the cell theory, cells arise from pre-existing cells, right? Okay, and cell theory was given by Schwann and Sheldon. Okay, and it is modified by Rudolf Virchow in 1955. The process by which this occurs is called cell division. Now, how cells arise from pre-existing cells, and this process is called as the cell division, right? Any sexually reproducing organism starts its life from a single cell zygote. Okay, cell division does not stop with the formation of the mature organism but continues throughout its life cycle, right? Okay, so cell division does not stop with the formation of the mature organism but continues throughout its life cycle, right? The stages through which a cell passes from one division to the next is called the cell cycle. The stages through which a cell passes from one division to the next is called the cell cycle. Okay, the stages through which a cell passes from one division to the next is called the cell cycle. Cell cycle is divided into two phases called interphase, a period of preparation for cell division, right? And mitosis, that is M phase, the actual period of cell division. So interphase, a period of preparation for cell division, and second is the mitosis, that is M phase, the actual period of cell division, right? The interphase is further subdivided into G1S and G2 phase, right? This is the interphase that is divided into G1S and G2. Now where we was at, so now what where okay, so the G1 phase is the period when the cell grows and carries out normal metabolism. Okay, most of the organelle duplication also occurs. So most of the organelle duplication also occurs during this phase, that is G1 phase. And S phase marks the phase of DNA replication and chromosome duplication. So S phase, that is synthesis phase, it marks the phase of DNA replication and chromosome duplication, right? So this is the S phase. And G2 phase is the period of cytoplasmic growth, right? G2 phase is the period of cytoplasmic growth. Mitosis is also divided into four stages, namely prophase, metaphase, anaphase and telophase, right? So where is the actual cell division occurs, right? Prophase, metaphase, anaphase and telophase. Now chromosome condensation occurs during prophase. Now the chromosome condensation occurs during prophase. During the prophase, right? Simultaneously, the centrioles move to the opposite poles. So during the prophase, the chromosome condensation occurs, and the centrioles also move to the opposite poles. The nuclear envelope and the nucleolus disappear, and the spindle fiber starts appearing. So nuclear envelope and the nucleolus, they disappear, and the spindle fiber starts appearing. Now metaphase is marked by the alignment of chromosomes at the equatorial plane. They align like at the equatorial plane. Okay, during anaphase, the centromeres divide. So during anaphase, it is a splitting, that is centromere splits, divide, and the chromatids starts moving towards the opposite poles, right? Once the chromatids reach the two poles, the chromosomal elongation starts. Nucleolus and nuclear membrane reappear, the initial stage, right? They reappear. This stage is called the telophase, right? This stage is called the telophase. The nuclear division is then followed by the cytoplasmic division and is called the cytokinesis. This stage is called the telophase. Right now, when the chromatids reach the two poles, the chromosomal elongation starts and nucleolus and nuclear membrane reappear. This stage is called the telophase. Now, the nuclear division is then followed by the cytoplasmic division and is called the cytokinesis. So, nuclear first division, and then there is a cytoplasmic division and is called the cytokinesis. Now mitosis thus is the equational division, because in which the chromosome number of the parent is conserved in the daughter cell. If the parent has the n, then the progeny has the n. If the parent had 2n, then the progeny has the 2n. So the chromosomes are same. Okay. Now, in contrast to mitosis, meiosis occurs in the diploid cells. In contrast to mitosis, meiosis occurs in the diploid cells. Like it's mostly occur in the diploid cells, which are destined to form the gametes, and meiosis is to form the gametes. Okay, not the mitosis. It is called the reduction division since it reduces the chromosome number by half while making the gametes. So when it makes the gametes, the chromosome number gets half. If parents are 2 and the progeny gets the n. Okay, in sexual reproduction, where the two gametes fuse, the chromosome number is restored to the value in the parent. In sexual reproduction, when the two gametes fuse, if it's n and n gets fused, the chromosome number is restored. It will be 2n, right? Same as the parent. Fuse, the chromosome number is restored to the value in the parent. Okay, now meiosis is divided into two phases, meiosis 1 and meiosis 2. In the first meiotic division, the homologous chromosomes pair to form bivalents. In the first meiotic division, the homologous chromosomes pair to form bivalents, okay, and undergo crossing over. Okay, now meiosis 1 has a long process. Which is divided further into 5 phases. Now itself the meiosis 1 has the long process and okay, long phase, which is divided further into 5 phases, right? These are liptutin, zygotin, pacatin, diplotin, and dikinesis. So liptutin, zygotin, pacatin, then diplotin and dikinesis, right? Okay, during metaphase 1 and in liptutin, what does happen? There is condensation of chromosomes, right? Then zygotin, there is formation of synaptinimal complex. Then pacatin, there is a recombination crossing over takes place in pacatin. Diplotin, what happens? There is a formation of X-shaped chiasmata, and in dikinesis there is a terminalization of X-shaped chiasmata. So this is the overall 5 steps, right? 5 phases. Now during metaphase 1, the bivalents arrange on the equatorial plate, same as metaphase during the mitosis. So during metaphase 1, the bivalents arrange on the equilateral plate. This is followed by the anaphase 1, in which homologous chromosomes move to the opposite poles with both their chromatids. Okay, now this is followed by anaphase 1, in which homologous chromosomes move to the opposite poles with both their chromatids. So homologous chromosomes, not the chromatids. Okay, each pole receives half the chromosome number of the parent cell. Okay, now the chromosomes are reduced in the meiosis 1. So each pole receives half the chromosome number of the parent cell. Like if it's 2n, then the progeny gets n, right? Now in telophase 1, the nuclear membrane and nucleolus repair. Now meiosis 2 is similar to mitosis. Now you see, mitosis n gives s. Now it's already been reduced, so n gives n gives n. We also want the n. If the parents are 2n, then we also wanted n. So after fertilization, they restored the chromosomes. I hope you conceptually understand. So meiosis 2 is similar to mitosis. During anaphase 2, the sister chromatid. Now the sister chromatid gets separated. During first, only homologous chromosomes separated. So chromatids, so centromere is not splitted. Okay, so during anaphase 2, the sister chromatid separate. Thus at the end of the meiosis, 4 haploid cells are formed. Now the exercise is, what is the average cell cycle span for a mammalian cell? I think 24 hours given in the NCERT. Distinguish cytokinesis from karyokinesis. Karyokinesis, it is a division of the nucleus, division of the nuclear material, right? The chromosome. And cytokinesis, it is a division of the cytoplasm, right? So describe the events taking place, and cytokinesis occurs after the karyokinesis is done. In many cases, cytokinesis does not follow the karyokinesis, so the polyploidy conditions occurs, that is multiple sets of chromosomes, mostly in the plants. Okay, you will learn it in the 12th class NCERT. So anyways, so describe the third question is, describe the events taking place during interphase. Now what's the things are taking place during the interphase? There is G1, there is S, and then there is G2 phase, then there is mitosis where actual cell division occurs. So there is also quiescent stage, that is G0 phase, where cell exist and do not go for further cell division until it's called on to do so. Okay, so all these NCERT lines, right? So describe the events taking place during the interphase. Okay, now what is G naught? That is quiescent phase of cell cycle, where when actual cell division is not, no, matlab cell is not ready for the cell division until it call on to do so. Okay, example is heart cell. Now fifth question is, why is mitosis called equational division? Because if the parents is 2n, the progeny will get the 2n. If the parents is n, the progeny will get the n. So this is why mitosis is called the equational division. Name the stage of cell cycle at which one of the following events occur. Chromosomes are moved to spindle equator. Chromosomes are moved to spindle equator. Okay, name the stage of the cell cycle at which one of the following events occur. Chromosomes are moved to spindle equator. This is anaphase. It can be anaphase 1 and it can be anaphase 2, right? Now it can be anaphase for mitosis, or it can be anaphase 1 for, not 2 mostly, because it's written that is chromosomes. It's not written the chromatids, I think. Now second is centromere splits and chromatids separate. Centromere splits. When does the centromere splits? It's in the anaphase 2, right? In the meiosis. Now pairing between homologous chromosomes takes place. Now pairing is means formation of synaptonemal complex. When it does it's in the zygote stage, right? Zygote stage. Okay, now crossing over between homologous chromosomes takes place. After the zygote there is a Pachytene stage where crossing over takes place, right? So this is it. Now describe the following. Synapsis, it occurs in the pairing of the two homologous chromosomes. It is synapsis, occurs in the zygote stage. Now bivalent, there is formation of bivalent, that is, two homologous chromosomes bivalent, that has 4 chromatids. Okay, now chiasmata, it is egg shaped. It's about to leave, like the two crossing overs when happen, it's about to leave. There is formation of egg shaped, at the end of the chromatids. It's still attached. Okay, draw a diagram to illustrate your answer. Now, how does cytokinesis in plant cells differ from that in animal cells? So how does cytokinesis in plant cells differ from that in animal cells? Okay. So you know in plant cells the cell walls is inextensible. So there is formation of plate from the middle of the cell, and then gets it centrifugally, and then it crosses the wall. So this is how cytokinesis takes place in the plant cells, while in the animal cells there is no such type of cell wall. So there is direct insertion centripetally and towards the center from the plasma membrane. So this is how cytokinesis in animal cells takes place. Now find examples where the 4 daughter cells from meiosis are equal in size and where they are found unequal in size. Find examples where the 4 daughter cells from meiosis are equal in size and where they are found unequal in size. So in humans, 4 daughter cells from meiosis are equal in size and where they are found unequal in size. Okay. Distinguish anaphase of mitosis from anaphase 1 of meiosis. Anaphase has no splitting of centromere, right? But no. Okay. Distinguish anaphase of mitosis from anaphase 1 of meiosis. Anaphase both has a separation of chromosomes, right? Now list the main differences between mitosis and meiosis. It is a equational division, it is a reductional division. It is a equational division, it is a reductional division, right? What is the significance of meiosis? Meiosis, it helps. What is the significance of meiosis? It's the production of the haploid cells, right? And then there is a variability, then the genetic variability happens, and then there is repairing, no, not the repairing mostly. So this is how meiosis significance is. Variation occurs. Okay, then meiosis also it it restores, it makes chromosomes gets reduced, and it's necessary for the to restore after the fertilization. Okay, so this is all meiosis. Now discuss with your teacher about haploid insects and lower plants where cell division occurs, and some haploid cells in higher plants where cell division does not occur. So haploid insects, they show the mitosis, right? Haploid insects and lower plants where cell division occurs, and some haploid cells in higher plants where cell division does not occur. Maybe the permanent tissues in which there are some haploid cells in higher plants where cell division does not occur. Now, can there be mitosis without DNA replication in S phase? Can there be mitosis without DNA replication in S phase? Can there be mitosis without DNA replication in S phase? No, it does not possible, because the progeny has to get the DNA. Okay, until if it's not copied, it shouldn't be there, right? So can there be DNA replication without cell division? No. Analyze the events during every stage of cell cycle and notice how the following two parameters change: number of chromosomes and per cell, amount of DNA content per cell. Analyze the events during every stage of cell cycle and notice how the following two parameters change: number of chromosomes and per cell, amount of DNA content per cell. So in S phase it is doubled. Okay, number of chromosomes per cells is the same, but on the chromosome there is DNA that gets replicated, right? But on the chromosomes, but 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 on the chromosome, the DNA gets replicated. So the DNA gets, if it is 2C then it's 4C, but chromosome stays the same. If it is 2N then it's also the 2N. So this is it. So this is all your chapter, that is cell cycle and cell division.